today's ASB. Hello, everyone, and welcome to today's ASBMB Breakthrough webinar, Advancing Cross-Linking Mass Spec to Elucidate Cellular Networks. I'm Chen-Min Jiang, ASBMB's Assistant Director of Publications, and I'm very pleased to be moderating this session today. This webinar is part of ASBMB's Breakthrough webinar series, which is sponsored by the ASBMB journals. Specifically, today's session is brought to you by the Molecular and Cellular Proteomics, MCP, one of our esteemed ASBMB journals dedicated to advancing proteomics in both basic and translational research. Before we dive in, a quick reminder, you are welcome to submit questions at any time using the Q&A feature at the bottom of your screen. We'll address them during the dedicated Q&A session after the presentation. This webinar is being recorded, and the recording will be available on the ASBMB website. It's now my pleasure to introduce today's speaker, Dr. Lan Huang. Dr. Lan Huang is a professor of physiology and biophysics at the University of California, Irvine. Her research centers on developing novel mass spec-based proteomics strategy to study micromolecular protein complexes, especially within the ubiquitin proteasome system. She pioneered sulfoxide containing mass spec cleavable cross-linkers, establishing a robust cross-linking mass spec platform of elucidating protein interaction networks and structure topologies. Her lab's methodology has advanced a study of the protein-protein interactions in living cells and led to patented commercially available reagents. Recently, her lab has applied cross-linking mass spec technologies to clinical samples to uncover disease-relevant protein networks. Without further ado, please join me in welcoming Lan. Lan, the floor is yours.
Thank you. Can you see my screen okay? Okay. Thank you very much for the introduction and the invitation to speak here. It is my great pleasure to share with you the work we have done on studying protein-protein interactions. So my lab focuses on developing mass spectrometry-based approaches to study protein complexes and including many different aspects such as subunit composition, post-translation modification, interaction, and structure and function. So we put a particular effort on studying protein-protein interactions due to their importance in regulating almost all biologic processes. As shown here, using a proteasome as an example, protein-protein interaction are critical for the formation and the assembly and the disassembly of the proteasome complexes under different conditions. And also it's important in controlling its activity and subcellular localization, as well as gene transcription. And also it's impacted by multiple factors, such as post-translational modifications. So the nature of the protein interactions is very complex. They can change with strength, duration, and they are also with temporal and spatial control. In recent years, it has been recognized that abnormal protein-protein interactions have been involved with the disease development, and the protein interaction interfaces have been recognized as attractive targets for drug development to improve therapeutic, for the disease treatment. So systematic analysis of protein-protein interactions will not only help us to understand cell biology, but also to provide new targets for improved treatment. So there are many different methods have been developed to study protein-protein interactions on the global scale. In particular, the affinity-based method have been very popular, and as shown here, that in the cells, if you have expressed tagged bayet through the affinity purification coupled with mass spectrometry, you can generate the bayet and prey interaction network. And in order to capture the transient protein interactions with a spatial resolution, proximity labeling methods have been developed in recent years and become very popular. So these allow you also through the labeling and affinity purification to generate a similar kind of network. These methods have been widely used and have provided a large amount of information for our understanding of protein interactions in cells. Although they are very effective and they do require the cell engineering by expressing tagged bayet, and also that these methods cannot distinguish direct interactor from indirect interactors. For any affinity-based purification, you often will have large backgrounds. This requires proper bioinformatics tools to filter out the non-specific interactions. So in order to address the shortcomings of those methods, we have focused on developing cross-linking mass spectrometry to study native protein-protein interactions. So as shown here, that cross-linking mass spectrometry requires the application of a cross-linker to allow the formation of covalent bond within protein or between two interacting proteins. And those cross-linkers carry at least two functional groups. So they often refer to as homobifunctional or heterobifunctional cross-linkers. And this allows us to capture protein interactions and through the chemical reaction between the proximal residues within the protein interaction interfaces. Through the identification of cross-linked peptides, then we can determine the protein interaction identities and also provide the physical context to elucidate the structure. And so for the cross-linking mass spec analysis, and the goal is to try to identify cross-linked peptides and to define the protein interactions with the identity and also the physical context. However, there are inherent analytical challenges associated with the identification of cross-linked peptides. As shown here, there are multiple different types of products that can be formed during the cross-linking reaction. And this, after digestion of the cross-linked proteins, it can result in the production of the interlinked peptides with two different peptides linked through the cross-link. And it can also generate this intralinked peptide or the dead end, which means only one end touched the amino acid, the other end actually is hydrolyzed or without reacting to other residues. So due to this complexity that it is often that the interlinked peptides are very low abundant. And these interlinked peptides, they are most structurally informative to allow us to determine the protein interaction interfaces. And due to the presence of the two pieces of the peptides, it causes the complicated fragmentation during mass spec analysis and lead to difficulty in database searching, as it will have the database expansion. So in order to facilitate the cross-link peptide identification, and we have focused on developing novel cross-linker reagents. In particular, we have developed the novel sulfoxide-containing MS-cleavable cross-linkers. So to talk about this, the first one actually we developed is called diisocyanate sulfoxide. And this cross-linker in short called DSSO. And this one consists of two reactive groups, the NH ester targeting primary amine, especially the lysine groups. And the key here is the introduction of a central sulfoxide. And due to the presence of this group, then we introduced two MS-cleavable bond between the sulfur and the carbon. And so this bond is much weaker than the peptide backbone. So doing the mass spec analysis for the cross-linked peptide, and if you can see here that when you form a cross-linked peptide detected in stage 1 mass spec, you can measure the peptide masses. And then in stage 2, we can use low energy collision-induced dissociation to physically separate these cross-linked peptides. And this allows us to subject them to the stage 3 mass spec, and then they can be individually sequenced. And these individually sequenced peptides can be subjected to conventional database searching tools. And through the integration of MS1, MS2, and MS3 data, we can unambiguously identify the cross-linked peptides. So after we developed the DSSO, we also developed other reagents. In particular, our goal is try to be able to map protein interaction in cells. And so we therefore developed this cross-linker called DSPSO, and which referred to the alkyne or azide tagged DSPSO. So these cross-linkers have several unique features. First of all, they are still homobifunctional cross-linker targeting lysine residues. Second of all, they actually carry the sulfoxide group, introducing the MS-cleavable bond. And importantly, we also introduced an enrichment handle. So this allows us to, using affinity-based approach, to isolate the cross-linked peptide to enhance the detectability and identification of the cross-linked peptide. So in order to facilitate the affinity purification, we also add the acid cleavable side. So this allows us to remove the enrichment handle after purification. So as shown here, this is the MS3 analysis of DSPSO cross-linked peptide. So doing MS1, we can measure the peptide mass, and doing MS2, they can, the collision-induced dissociation will trigger the characteristic fragmentation of DSPSO cross-linked peptides and generate these predictable pairs, which can be subjected to MS3 analysis. And through the MS3 sequencing, we can unambiguously identify these individual peptides. And through integration of this all MS data, then we can determine the cross-link between these two peptides. So once we generate these cross-linkers, then we have applied them to the intact cells. The first example I'm showing here is that we applied DSPSO cross-linking to HEC293 cells. And so this is a general workflow. The first step is the in-cell cross-linking. And afterwards, then we will through different solution or buffers to extract the cross-linked proteins. And then we would digest the cross-linked proteins to generate cross-linked peptides. And in here, there's a mixture of peptides, including cross-linked products as well as linear peptides. So in order to enrich cross-linked peptides, we used click chemistry-based conjugation and then affinity purification. So all different types of cross-linked peptides will be enriched through this process. So in order to isolate the most informative interlinked peptides, then we subjected it to size exclusion chromatography based on their peptide size. So this way we can focus on analysis of the interlinked peptides. And then using the advanced mass spectrometry and through the LC-MSN analysis, we can identify the cross-linked peptides, and this allows us to generate the cross-linked maps as well as the interaction maps, and the information can be used for structural modeling. So for the HEC293 cells, we generate a large amount of interaction data, as shown here that the LC-MSN allows you to identify cross-linked peptides and generate residue-to-residue linkages and to determine interaction interfaces, and then to deduce the protein-protein interactions. So through this analysis, we can generate a larger number of protein mapping, and this allows us to compare to the shotgun proteomics proteome to indicate that cross-linking mass spec can cover multiple subcellular compartments. And the information allows to generate not only the global interaction network, but also can zoom in to derive the pathway-specific interaction networks. So this work has established the solid foundation for us to further apply this technology to map the protein interactions in other samples. So as I mentioned before, that abnormal protein-protein interactions have been implicated in different disease. And the disease-triggered rewiring has been linked to, the development of the different disease stages. So in order to understand the disease-specific interactions, we have applied the DSB-SO cross-linking mass spec strategy to different clinical samples. So as shown here, we have applied to breast cancer patient-derived xenograft models, and this work has been published last year. And we also have applied to this strategy to tumor tissues from patients, and this is a project within the Clinical Proteomic Tumor Analysis Consortium. And so today I will share with you some of the unpublished work we have done from this CPTAG project. So we have analyzed different cancer types. And so here is the distribution of different cases and deaths for the top 10 cancers. As shown here, these pie charts reflect the incidence and the mortality from both sexes. And as shown here, that the stomach cancer is the top 5 cancer occurrence, and also for the deaths within the different types of cancers. So we have one of the project is to try to study the protein interaction in the gastric cancer, and to provide molecular signatures to hope to allow us to contribute to the understanding of gastric cancer, and also potentially to identify molecular markers for the diagnostics and the treatment. So before I show you the data, I also wanted to mention the complexity of the formation of the cancer, and also, the outcome may also be difficult to understand. And as shown here, there are different risk factors can trigger the gastric cancer. They are non-modifiable factors, and they are modifiable factors. So you can see that this complex formation and will generate the difficult, have a complex mechanism underlying this disease development. So in order to contribute the understanding of this cancer progression, we have tried to use the DSB-SO cross-linking mass spectrometry strategy. So in here, due to the large number of samples, so for this particular project, we obtained 190 samples, including 150 tumors and 40 normal adjacent tissues. And in order to characterize the protein-protein interactions within these samples, we actually employed TMT-based multiplexed quantitative approach. So as shown here, that those tumor samples have been collected, they are quite pulverized, and we carried out the DSB-SO cross-linking, and then we lysed the proteins and doing the digestion, and also that through the click chemistry conjugation, and then we used the TMT labeling, and for us to mix the samples, and then for the multiplex analysis. Due to the complexity of the sample, we actually also applied 2 additional peptide fragmentation techniques, including size exclusion, as I mentioned before, but also coupled with a high pH reverse phase, the fractionation, to further reduce the complexity to improve the analysis dynamic range. And once we collected the samples, then we subjected them to the multi-stage mass spectrometry analysis. So as shown here, again, that we have MS1, MS2. One thing I would like to point out is during the MS3 analysis, the TMT reporter ions can be observed in the MS3 spectra. And so then in here, you have two portions. The reporter ions allow us to do the cross-linked peptide quantitation, and then the sequence ions allow us to identify the cross-linked peptides. So this is a perfect coupling with the TMT-based strategy. So once the data is obtained and the cross-linked peptide identified, then we can use the information for the addition for the cross-linking mapping and structure mapping, as well as network mapping, and to understand the disease specific protein interactions. So today I will focus on the analysis we have done, use the stage combined samples. So as shown here, we actually, in this cohort of samples, we have 4 different pathologic states, and you have stage 1, stage 2, stage 3, and stage 4. And then we also have the NAT tissues. And so what we have done is that in addition to individual sample analysis, we combined these samples into different stages, is try to give us a global view of how the stage impact the protein-protein interactions. So the combined sample also labeled with the TMT for the multiplexing quantitation. So for this set of analysis, we actually have identified around 3,000 linkages, and these linkages have a good percent of the interprotein and also intraprotein interactions. After the identification, then we also quantified those cross-linked peptides, and based on the log2 of the cross-linked peptide intensity, this box plot indicated that the median and the distribution of the intensities, they are very similar across the biological replicates, and also that the overall has a good normalization and high reproducibility. Then with this data, we also try to understand what kind of information is enriched. So we used the Go enrichment analysis, and here shows the cellular compartment enrichment in the XL proteome. And so we compared the gastric data with our previously published breast cancer PDX model data. And you can see that the top enrichment are quite similar. So indicating that there are some, the common cellular components are present in these, the cancer samples. So to further understand whether these data can be well separated to determine the stage-dependent interactions, we performed the principal component analysis of the cross-links across different stages and replicates. So as shown here, that each group of the replicas, they have grouped pretty well in close proximity, and suggested the high reproducibility during the data analysis, during the experiments, and the fact of the separation of different clusters, and especially the separation of the native adjacent tissues from different tumor stages, as well as the separation of different stages, indicating that there's a the cancer-induced rewiring of the protein interaction network. And this also suggests that the protein interactions actually is stage dependent. So to further understand the nature of these interactions, we plotted the abundance changes and compare the tumor versus the net. as shown here that while majority of the cross-linked peptides did not change, but there are a group of cross-linked peptides showed either upregulated or downregulated changes. So the question is whether these cross-linked changes are due to the abundance changes in the protein expression in different tissues. So then we plotted this comparison between the protein expression changes versus cross-linked peptide changes. As you can see here that not always the cross-link changes correlate with the protein expression changes. So sometimes when you don't have the protein expression changes, you have a cross-link changes indicating interaction changes. And then obviously when protein abundance increases and you can observe some protein interaction increases as well. And this also correlates with the downregulation of the protein expression levels. So this plot tells us that it's not a simple correlation between the the cross-linking changes with the protein expression levels. So that means that protein-protein interaction rewiring is much more complex and can provide additional information beyond protein expression levels. So here are some examples to show you the correlation of the altered cross-links with protein abundance and the structure. So this particular protein and expression has been implicated with poor prognosis in the gastric cancer. So as shown in the literature that this protein is downregulated in the tumor samples. And this is also being validated by our proteomics data, as well as showing that there's a correlation with the downregulation of the protein-protein interaction detected here. And this cross-link correlates with the known structure pretty well. And then another protein showing here, and actually have an opposite effect, and why the protein expression is actually downregulated in the tumor samples, but their interaction shows upregulation in the tumor samples. And this cross-linking also correlates very well with the predicted structural model shown here. So this further indicates the complication and also the additional information can be provided by protein interactions. So in addition to the intraprotein interaction, this shows the example of the interprotein interactions. And so this is the interprotein interaction between TEFM and the PGD. And as shown here, these two proteins actually did not have significant changes in the protein abundance across different samples, but their protein-protein interaction has been significant induced in the different stages of the tumor samples. And in addition, there's another protein-protein interactions identified between AN01 and the PDE9A, and a similar situation has been observed here. And one interesting aspect is that the PDE9A actually expression has been linked to the survival with the different type of cancers. However, this particular protein was not identified in the whole proteome analysis. So indicating that the cross-linking analysis could reveal additional proteins which are missed during the shotgun proteomics. And so these cross-links, these interactions are novel interactions, and using predicted model, the distance indicating is beyond the desired distance restricted by the cross-linker, suggesting maybe there's conformational changes during the different disease states. So with the information, and we are able to construct different protein interaction networks, as shown here, that we enriched those interactions at different cellular processes, and those processes carry out interactions. They are either upregulated or downregulated and correlate with different disease states. So this set of data and also the study provide resources for us to further understand the protein interaction linked to gastric cancer progression. So one thing I would like to mention is that in addition to the whole proteome analysis, in collaboration with Professor Aaron Witter's lab at UCSF, And we have started to use cross-link mass spectrometry coupled with membrane protein enrichment to study the structural surfaceome for cancer therapy. In particular, his lab is interested in acute myeloma, leukemia, and try to identify the disease-specific conformation and use that as the therapeutic targets. And in this first work published in Nature Cancer, and using DSSO cross-linking actually identified the active conformation of the integrin beta 2 with integrin alpha L. And in here, you can see that it shows the distance of these two cross-links exceeding the required the distance threshold and indicating the open conformation. And then his lab confirmed this conformation present in the diseased cell lines. And then this active conformation can actually be used for eliminating AML cells without targeting the normal cells. And this kind of closed and open conformation can be replicated through biochemical manipulation through manganese binding. So recently, we also coupled this with other cross-linkers, and hopefully in the future, we can present the information to you and to further, enrich the membrane protein, the interactions. So in addition to the lysine to lysine reactive cross-linkers, we also have developed other chemistry reactive cross-linkers to expand the protein-protein interaction and the coverage. So as shown here, we developed a cysteine reactive MS-cleavable cross-linker, and this one is called DBRSO, and carries a brunoacidamide, which targeted the cysteine residue, and also still carries the sulfoxide group as the MS cleavability. And in comparison to the lysine reactive identified the cross-linker proteome, and you can see that there's additional proteome identified, so expanding the coverage of the entire proteome. And also due to the low abundance of cysteine, we actually can also cover different aspects of the low abundance protein-protein interactions. So this study indicating that a multi-chemistry cross-linking approach can actually broaden the spectra of the protein-protein interactions. So recently, we also developed MS-cleavable cross-linker for mapping the multimeric, the protein-protein interactions. And the reason we wanted to do so is because we wanted to facilitate the structural characterization of heterogeneous protein complexes. As I mentioned, the current existing cross-linkers, they are bifunctional. And as shown here, if you have a different type of combination of trimeric complexes with the bifunctional cross-linkers, you can generate those pairwise interaction. And only the presence of pairwise interaction, it will be difficult for us to distinguish those different types of combination of this trimeric complex. If we have multi-link cross-link identified, then you can allow us to determine the presence of AAAB, ABC, or ABD. And this can be also differentiated from other types of trimeric protein complexes. So in order to do that, we actually develop a novel trioxane-based MS-cleavable cross-linker for mapping the multimeric interaction. So this cross-linker is called TSTO, represents tris-succinimidine trioxane. As shown here, the key structure is this trioxane. The beauty of this structure is that during mass spec analysis, this allows us to concurrently release all of these cross-link arms. So in here, we have 3 reactive groups, so we can target 3 lysines. And this allows us to capture different type of cross-linkers. And this MS cleavability introduced by the TSTO allow us to identify these cross-linked peptides unambiguously. So in here that, as I mentioned, it will capture different types of the cross-linked residues these are tri-links, and these are actually tri-links between two different peptides, and these are bi-links within two different peptides. So this particular linker is also cell permeable and allow us to do intact cell analysis like DSBSO. So we not only capture large number of cross-linked peptides mapped to existing structure, but also generate the comprehensive interaction network. But the most interesting part is that due to the capture of the tri-link cross-links, it allows us to identify the multimeric protein complexes present in the cells. As shown here, this is a trimer formed in NME2 protein, and here is another trimer formed between among these proteins. And so here is another trimer protein complexes. And so this indicated that the TSTO cross-linking allow us to better characterize the homomeric oligomers. And another important aspect is that due to the tri-links and using proteasome as example, we have demonstrated that it can improve the accuracy and precision of the structure modeling. As shown here, this is the data compared with the DSSO cross-linking, and then you can see that the precision, the accuracy and the precision are significantly improved with these tri-linked cross-linked peptides. So through the years, we have developed almost 20 cross-linked peptides, and I talked to you about the DSSO and also many focused on DSBSO application in protein-wide analysis, and touched upon DBR-ASO and TSTO. So we also developed other cross-linking reagents, and for example here, SDASO targeting lysine residue and non-specific residues. So all this is try to further expand the protein interaction coverage. And with these reagents, we have applied to different systems. These allow us to generate site-specific PPI interaction networks, and also to allow us to determine condition or disease-specific interactions, as well as the conformation. And I didn't have time to talk too much about the structural modeling, because the cross-link can be used as distance restraint for facilitate the structural validation and modeling. So in summary, I hope I have showed you that to address the current challenges, we actually have developed different cross-linking reagents to facilitate the identification of the protein-protein interactions, and then developed different analytical strategies to further expand the protein interaction coverage and the depths of the interaction studies. And we have applied to different biological and clinical samples, and these strategies can be used for protein complexes, subcellular organelles, and tissues, and different types of patient samples. So at the end, I would like to thank the people in the lab, especially Fenglong Zhao. He has done all these cancer studies, and with the help from Clinton Yu, and all these people contribute to the development and the application of the cross-linking technology in our lab. And those are the previous members contribute significantly to this research I presented here today. And also, I wanted to thank my longtime collaborator, Scott Rigonowski, for the development of MS-cleavable cross-linkers, and thank the Al Burlingame Lab for the support of Protein Prospector, and Andre's lab for the structure modeling, and Arun lab for the collaboration for the membrane, the cross-linking study, and thank Hui Zhang's lab and Bin Zhang's lab for the CPTAG, the project, and also the entire team of the CPTAG, as well as the Ian and Rosa in Thermal for the collaboration, and other people for different types of the contribution for the collaboration, and also for thank NIH for the funding, and again the CPTAG, the team, as well as thank you for your attention. I'll be happy to answer any questions.
Thank you, Lan, for the insightful presentation. So we have now about 10 minutes for the questions. So we have a lot of questions in the Q&A. Hopefully we can cover them all. So the first question, are the cross-linkers that you are using, first one. Oh, are the cross-linkers that you are using membrane permeable, or are you using permeable cells for the in vivo cross-linking?
So the cross-linkers we use, the DSBSO and the TSTO, they are membrane permeable. For all of the data showing here, the cross-linking can be applied for the intact cells and also intact tissues, which I didn't have time to talk to you about.
Okay, thank you. The next question is it possible to identify de novo peptides from these cross-linking methods?
So it is possible because, as I mentioned, for our cross-link identification workflow, we actually use MS3 type acquisition and analysis. And so in there, you using, you sequence the individual peptides. So it's very similar when you do protein, like the protein identification from de novo sequencing. So if you have the MS3 data, potentially you could use those de novo sequencing software. The only thing is that the targeted residue, such as lysine, you will carry a defined modification on there. Which is also similar to a post-translational modification, and you can do the de novo sequencing on that aspect.
Thank you. So the next question, is there value in using DSBSO derivatives that have central spacers covering a range of different lengths?
Yeah, so this is a very interesting question. So during our development of different cross-linkers, we actually explored different lengths of the cross-linkers. And what we have found is that the lens actually will impact the range of interactions you capture, but also actually impact the cross-linker structures in a way, their flexibility and whether they could like a folding your bend. So if it's too long, one thing is that it can actually reduce the reactive group activity, and also that it allows the linker region to be too flexible, then the distance becomes less informative. So based on our experience, we feel like from, like a 7 to 14 angstrom is actually pretty good length, and they actually capture the cross-linker residues within very similar distances.
Thank you. So the next question, in the multiplex experiments that you described, what's the rationale for mixing the samples prior to mass spec analysis?
So we try to use the entirety for the labeling because we try to prevent the variability. And so this way, by, so because we enriched first, we actually enriched the entire cross-linked peptides first before we do the label. And this technically is supposed to be only labeling the cross-linked products. And so in here, we actually, this labeling will allow us to focus on the cross-linked peptides and also allow us to quantify in addition to the interlinked peptides, but also the interlink and dead-end cross-linked peptides. And even though we do not use those linear peptides, these cross-linked linear products to determine the protein-protein interactions, but this information is useful for to compare to the protein abundance changes.
Thank you. So for next question, for all of tumor samples at different stage, were they from the same cell type origin, or are these of heterogeneous origin?
Yeah, this is a very good question. So for this set of experiments, and this has a heterogeneous population. So it would be nice in the future that we will focus on, different cell types, and then to understand better of their, the protein network associated with the disease.
Mhm. Thank you. So next question, to determine the clinical stage, how much tumor size will be required for this cross-linking mass spec technique? In other words, will it be invasive for the patients?
No, so these samples are collected by NIH, and so the amount is not that much because when you try, this is another reason we use multiplexing because you actually the input can be reduced. So for the samples we have done for this set of experiment, it ranges from 15 to 30 mg tissues used. So it's a small plug for this analysis. So if for this particular example I showed, we used 11-plex. And so recently we started to use the 16-plex. And so for that, we can, further reduce the amount required for the tissues.
Okay. So next question, what's the availability of cross-linkers? Are they now commercially available?
Yeah, so about seven crosslinkers are commercially available, including the ones I talked here today. So the DSSO has been widely used, and DSPSO also now being used by many groups. So these two reagents definitely they are available, sold by multiple companies, and some other reagents only available by Sigma. DSSO, DSPSO, they are available through Thermo, Sigma, and other companies. And so hopefully the other reagents will become available soon.
Okay. So we have three more. Maybe we can just get one more. So how do you set up the distance constraint for TSTO crosslinker?
So the TSTO, the design is equal arm length. And so the distance calculation, because it's it's a three-way like a triangle. So actually, when we define the distances, you have to calculate the distance between the two residues, and then to determine the distance for the structural modeling.
Um, Thank you. Thank you, Lan. And thank you, everyone, for submitting such thoughtful questions. I'm sorry we cannot cover all the questions due to the time. And Lan, thank you for your detailed response. And now I would like to hand back to you for any final thoughts or key takeaways before we conclude today's session.
Well, thank you all for listening, and also thank you for all these wonderful questions. And because we are a technology lab, so today I presented mostly about the technologies in cross-linking mass spec to study protein interactions, and showed the application for different samples. So we actually also coupled this strategy with affinity purification as well for targeted protein complexes. So in a way, I feel like that the strategy has a broad application, and because of the availability of these reagents, from different companies, and I think that you could apply the same strategy and then to study protein interactions in the systems you are interested in. So we continue to develop new reagents just to try to have a suite of tools to allow us to be able to increase the in-depth coverage of the protein interactions. And the goal really is to provide detailed information and then contribute to systems structure biology. And so hopefully, because if you also use this strategy, then I think that the impact of this kind of technology would be more broadly showing the different applications. So yeah. So if you have any questions, feel free to let me know. And thank you very much.
Thank you so much, Lan, for your time and expertise today, and to all of you joining us. Be sure to keep an eye out for next month's webinar in ASBMB's Breakthrough Webinar Series, and as the final one of the year. And have a great day. Bye.
Thank you. Bye.